Hello and welcome to Antibody Engineering and Therapeutics Digital Week, brought to you by the organizers of the Global Antibody Events and Content Series. My name is Brianna Kosofrat and I'll be your host for today's session titled High-Throughput Screening of Twin Strep-Tagged Nanobodies using Next-Gen Biolayer Gator Pro System. First, I'll cover some quick housekeeping items. If you experience difficulties with audio or advancing slides, refresh your screen with F5. If you are experiencing other issues, hit the question mark button to receive assistance. At any time during the presentation, you can submit your questions into the Q&A window on the left-hand side of your screen. In 24 hours, you'll receive a link to watch the recording of the session. You can also download a featured resource in the resource list box on the right-hand side of your screen. Now let's begin by introducing our speakers. Today we have Fabian Moore, PhD. He's Vice President for Research and Development at IBA Life Sciences. And we also have Johnny Zhang, PhD. He's a research scientist with Gator Bio. Welcome, Fabian and Johnny. I'll now turn it over to you to begin the presentation.
Hello and welcome to the first part of the webinar titled Strep-Tag, a robust affinity tag platform. My name is Fabian and I'm the Vice President of R&D at IBA Life Sciences. We are founded in 1996 and are located in Göttingen, right in the middle of Germany, and have around 60 employees. We are ISO certified and have a worldwide distributor network, making our products available in 40 countries, and have customers in academia, biotechnology, and pharma industry. Our core platform is based on strep-evident mutants called Strep-Tactin and Strep-Tactin XT, and their specific interaction with a small peptide tag called Strep-Tag. Strep-evident is a versatile protein that has been widely used in biotechnology and life science research. It is known for its high affinity towards biotin, a small molecule that is essential for non-biological processes. Strep-evident is a tetrameric protein and can bind 4 biotin molecules. The interaction between strep-evident and biotin is one of the strongest known non-covalent interactions in nature, with a dissociation constants in the femtomolar range. To achieve this strong binding, the lid, here marked in red, closes after biotin binds into the biotin binding pocket. This strong binding, while being an advantage for many applications, is also one of the greatest weaknesses of the biotin strep-evident system, as this binding is virtually irreversible. We set out to generate the before mentioned proteins Strep-Tactin and Strep-Tactin XT, both mutants of strep-evident, which alter the affinities towards biotin and a small 8-amino acid peptide tag called Strep-Tactin. Like strep-evident, Strep-Tactin and Strep-Tactin XT are also tetrameric proteins binding to the peptide as shown here in yellow with a highly specific binding pocket, but very different affinities towards the peptide, with a rather low affinity in Strep-Tactin and a very high affinity in Strep-Tactin XT as shown in the right graph. The possibility to now combine the low affinity Strep-Tactin and the high affinity Strep-Tactin XT together with Strep-Tag2 or twin Strep-Tag, doubling the peptide sequence for the monotag, allows us to tweak the system from a micromolar, very low affinity interaction, for example for purification purposes, over a nanomolar interaction between Strep-Tactin and the twin Strep-Tag, and Strep-Tag2 and Strep-Tactin XT to a very high interaction between the Strep-Tactin XT and the twin Strep-Tag, which can be used for immobilization of the protein, purification and detection. Today we will focus on the interaction between a twin Strep-Tag protein and Strep-Tactin XT, but almost all of the steps I show you today are also applicable for all other combinations of target proteins and Strep-Tactin mutants. The first step of protein purification is the specific binding of our target protein to Strep-Tactin XT. followed by washing away unwanted proteins, and then releasing our target protein in a third step, specifically by the addition of biotin, which replaces the twin strep-tag protein inside the binding pocket, releasing our target protein. After the specific elution of our target protein, we can regenerate our reagents afterwards for further uses. This system is independent of the matrix you can use. You can have magnetic beads coupled to Strep-Tactin XT for screening purposes. You can couple Strep-Tactin XT to agarose beads to use drip columns for gravity applications, or FPLC columns for an automated, rather large-scale isolation of your target protein. The first look we are having here is the isolation of GFP labeled with a twin strep-tag from a whole lysate. As you can see, the overexpression is not too strong, and we have here replicant isolations of twin strep-tag GFP every time from a whole lysate. And you can see in the 9 samples shown the purity in a one-step purification process using our magnetic beads is quite high. A second application I want to share with you is the isolation of different nanobodies, also labeled with a twin strep-tag, which target the molecule CD45, where you can see that there are differences in yield, but we can isolate in parallel multiple nanobodies for further applications afterwards. And this process, as I mentioned before, can be upscaled as well. So after you decided to go for one of the target proteins, you can go, for example, in fermentation, and as you can see here in a colloid 10 liters, and we have a yield of 2 grams per liter of our target protein, a CD45 nanobody labeled with a twin strep-tag. And as I mentioned before, this is a one-step purification process where we already got 99% purity as shown in the gel on the right. And in the analytical HPLC, we also confirmed the purity afterwards. So after showing that the isolation of the protein works quite well, we also wanted to share some data for the regeneration of the FPLC columns. And for that, you can see here different cycles run afterwards of the regeneration using sodium hydroxide. And depicted here are cycle 10, cycle 25, cycle 50, cycle 75, and cycle 100 of regeneration. And then, as you already can see in this graph, that they are almost identical. For analysis and quantification, we always use the dynamic binding capacity at 10% breakthrough and compared it to our first run. So it's always depicted now from the first run and then seeing the decline of our functionality. And we can see that both 0.1 molar sodium hydroxide as well as magnesium chloride can be used 100 times without any decline in functionality of our columns and the FPLC, whereas a harsher condition using 0.5 molar of sodium hydroxide will lead to some decline, which is similar to the decline you would expect, for example, in a protein A column. Not only is the one-step purification a very fast way to isolate your target protein, which can be easily scaled as well, as I've shown before, it is also compatible with a lot of media. As I've shown before, all the data were E. coli, but it's similar in insect cell lines, mammalia cell lines, and also cell-free systems, making it very versatile. I did show you a short overview over all 4 steps of our purification cycle, from the specific binding of our target protein over washing away and the high purity gained by that of other proteins, to the specific release of our target protein and the capacity to regenerate our columns 100 times without any loss in functionality. But we want to also illustrate that the application of the twin strap tag doesn't end with the isolation of your target protein. We talk always about a platform application, and we want to illustrate in this graph that it can not only be used for the production of your protein, the purification of your protein, but you can also generate in parallel staining reagents for FACS analysis within minutes. You can use it for the direct enrichment of cells and vesicles, or go more into protein analysis like Western blot, ELISA, SPR, and especially BLI analysis, where Johnny from Gator Bio is going to share some data how this really can impact your research. With that, I want to thank you all for your attention, encourage you to contact us if there are more questions or ideas, and now I'm going to hand over to Johnny.
Okay. Thank you, Fabian, for the fantastic first part of the webinar. And during the second part of the webinar, we will be introducing and talk about how we take these twin strap tag expressed nanobodies and apply our Gator Pro instrument to do a high throughput screening to find out what's the best nanobody. The agenda of the second part of the webinar is to first introduce who is Gator Bio and what is the technology behind our instruments. And moving on, we will be focusing on the main part of the study, which is the nanobody discovery research and how did we first design a biosensor for this application, especially to bind the twin strap tagged proteins using the Strep-Tactin XT proteins provided by the IBA. And moving on, we will use this biosensor in combination with our instrument to do a full panel kinetics and quantitation analysis for each nanobody. So, who are we? Well, we are label-free, the instrument company, and what that means is we utilize the technology called BLI to study the molecular interactions, and in particular, the quantitation and the kinetics of the biomolecules. And one of the main applications of a technology is to looking at the antibodies interactions. And that allows us to do both the quantitation, kinetics analysis, and the epitope of the antibodies. And of course, we are not limited to antibodies. We can also measure the small molecule bindings. And recently, we also branched into the gene therapy market where we can look at the AAV quantitations, both in crude and purified samples, and also the empty versus full ratios, and many applications beyond. We're headquartered in Palo Alto, California, and the company is created by the original team who developed the BLI technology 15 years ago. So what is BLI? Well, it stands for Biolayer Interferometry, and it actually happens in our everyday life. For example, a soap bubble that you can see here with different colors on the surface is actually because of the thickness of the soap film. And the soap film will interact with the light wave. And when it comes back to your eye, it interferes it because of this film, and the pattern shifts. And the wavelength shifts, changes the color, and then we can see that through this interference pattern. And how did we use this principle? Well, we take a biosensor, which is a glass rod, and at the end of it, we couple a light source and a detector to collect the light wave that comes back. And the light waves will travel through the glass rod, enters the tip of our biosensor where we have our molecules immobilized. And if there is any binding at the tip of the probe, the light wave will get interfered because of the presence of these molecules. And then when it comes back, we can see the phase shift of the light wave. As you can see here, that as time goes on, the molecule binding onto the top of the curve, the top of the tip, and then we can see the shift of our signal. And from there, we can generate the signal response to the time, which indicates the binding. And of course, when the molecule dissociates, the signal will go down, and then we can measure the dissociation. And from both the association and dissociation of the molecule, that gave us the kinetics constant. And these constants are important, such as the K on, K off, and the KD that you might have heard of. These constants tell us the interactions between the biomolecules. And we offer a range of products to support this technology. Of course, the main instruments are the Gator Prime, Plus, and the Pro, each optimized for different applications and user needs. And we offer a huge catalogs of biosensors and consumables to make your research easier, and the software accompanied by it. And what did we make the BRI technology even greater is summarized in a few points. And the main point is actually we have a new proprietary surface chemistry that are really optimized for the molecular bindings to reduce the backgrounds and increases the actual molecular interaction signal. On top of that, we have also a brand new optical layer, which allows a enhanced interference that actually also amplifies the signal on top of that to make our instruments more sensitive. And because of that, combination with the chemistry and the new optical layers that actually allow us to compatible to even wider range of the biomolecules, and including molecules such as the lipid nanoparticles, and of course we have our AAV solutions. So we are not just limited by small molecules and the proteins. So we can actually go much beyond that. And of course, the workflows and the software are optimized for easier and more friendly user-friendly. And in combination with that, we have a range of biosensors or kits that you can use right out the box. So how do we support the biotherapeutic research? And it actually goes from the early discovery to all the way to the manufacturing QCs. And for example, during the antibody market, we mainly supports the early discovery where you can choose and then screen for the best binding antibody. And we can optimize these best binding antibodies and do a more detailed kinetics analysis on the selected candidates. And from there, we can even support the process scale up, the manufacturing part, by quantitate these, produce the antibodies and QC these antibodies. And although it sounds very straightforward from the nano antibody market and to apply that directly to the nanobody market, however, there's still a few things that I wanted to point out that are still challenges to do currently. And while you're talking about the nanobody discovery and the whole pipeline discovery process, it's actually there's a more additional criteria that are different to the antibody. So current challenges that we all know in the traditional antibody market is the trade-off accuracy with throughput. So you can imagine the more accurate methods that you choose to look at your antibodies, you also lose the throughput. A lot of methods that are highly accurate are impossible to do with hundreds and thousands of samples. And of course, SPR analysis has been the gold standard in the field for many, many decades, but we still need efficient screening tool because to run these SPR analysis on hundreds and thousands of samples is unfortunately not possible. Of course, not to mention the hands-on time, regardless of the SPR or ELISA, you will require highly skilled, trained personnel to operate each assay. So with that, we really need some sort of an automation and with the high throughput, without the trade-off the accuracy to really meet the needs in the antibody market. And in addition to that, we have a few things that are unique to the nanobodies. For example, sometimes or oftentimes, in fact, that these nanobodies already had its sequence and we were just recombinantly produce these. And in this case, when we are doing kinetics analysis, these nanobody has to have a different tag compared to its antigen. You can imagine if both of your nanobody and the antigens are expressed in his tag, you wouldn't be able to immobilize one of them while looking at the other antigen binding onto the nanobody, simply because they share the same tag. And And the tag itself has to be very specific. In this case, if you want a quality kinetics data, when you immobilize your nanobody onto the sensor, it has to be immobilized with a very, very tight chemistry. So in this case, a very tight immobilization tag. And as Fabian mentioned, that the strep tag system, so the twin strep tag and the Strep-Tactin XT actually fits this criteria. And of course, because of the size of the nanobody, these are 15 kDa proteins, and therefore they're 10 times smaller than the traditional antibody. So therefore, we would require a much more sensitive techniques to be able to get a quality data. So how do we address these particular challenges in the antibody market, but also the specific things for the nanobody market? Well, first is our instrument Gator Pro System that are optimized for high throughput analysis. This instrument is designed for screening a large panel quantitation, kinetics analysis, and of course the epitope binding. And you can see here, this is actually the only BI currently on the market that offers 32 dedicated high frequency channels. So each channel represents a sensor, and they're not being multiplexed, meaning one sensor is not being shared to look at multiple wells. It's actually one high frequency channels with each biosensor. That allows us to achieve accurate kinetics each time when we run these samples. And of course, the instrument is designed to have minimal hands-on time for fully automation assay. Two quick examples of what this instrument can achieve, which is first is the quantitation part. Here we demonstrated that within 2 hours, we were able to quantitate 1,152 samples. And for the kinetics and epitope binding part, we showcase with 32 channels, we can do 32 by 32 epitope bindings, all completed within 8 hours. So that's the machine that essentially solves the throughput and accurate analysis screening tool part of the challenge. And the second part is actually how can we apply this to a nanobody screening situations. And for that, like I earlier mentioned, it has to have a different tag other than the commonly used his tag, and mostly because we need something much tighter immobilization method. So in this case, we engineered a biosensor that utilized the Strep-Tactin XT interaction and the twin strep tag. Currently, it's possible to do this. Unfortunately, it's a little bit labor intensive because each time before you start your run, you actually need to aiming couple these Strep-Tactin XT proteins onto your sensor. And that takes about 25 minutes before each of your experiments. And not only to that, between the day-to-day and the reagent lot-to-lot, it could very change the immobilization efficiency. So therefore, you would have some variation between the run. On top of that, because of the amine coupling method, the density of your protein is in comparison lower than if we design a specific sensor that are made with these proteins. So in this case, we use our internal chemistries that actually made the probes that are in high density with the Strep-Tactin XT proteins immobilized. And on top of that, you don't need to go through the amine coupling methods. So you can use our probe right out of the box. And we also ensure the consistency between each of your runs. And because of our chemistry and the unique optical sensors, it really adds up you would have 3 times higher signal that allows you to see small proteins like the nanobodies. So here you can see an examples of this sensor being used. Here we have a first step, which is the baseline and the buffer. And the second step, you can see the binding of the twin strep tagged GFP. And this part is the binding, and here you can see the dissociation, and this showcases how tight the twin strep tag with the sensor. You can see that the KD is indeed around the picomolar range, meaning it would be a fantastic immobilization tag for any kinetics run. And we take this sensor, and you can go beyond just the loading step. We can also measure after you load your twin strep-tag proteins, and then we can look at the subsequent protein interactions, which give us the kinetics binding. And in this case, we show that a twin strep-tagged IL-17 A and F protein, it's a heterodimer. We can load this twin strep-tagged proteins onto the sensor. You can see here the step that it increases as the sensor dipped into the well that presents with these proteins. A quick baseline to equilibrate the system, and then we dip the sensor into the IL-17 RA, the receptor part, and you can see the receptor binding and the association and dissociation to therefore get a very quality kinetics analysis between the pair. So now we have the sensor, and now we have the machine that allows the high throughput analysis. And how are we going to approach this nanobody screening discovery study? So we have anti-CD45 twin strep-tag expressed nanobody. And just a quick reminder, what are these nanobodies? So nanobodies are actually derived from camelid antibodies. So in for example, in camel, their antibodies shaped quite different from what we know. They have this variable regions, just single domain expressed like this, unlike our FAB region. So what you could do is just take, similar to how people use the FAB as one of the a new target, but we can also just take the single domain part, and that is what we call the nanobody. And what's our target? Well, the target in this case is CD45, which is a receptor called, it's highly conserved receptor, protein and tyrosine phosphatase. And it's actually commonly used for therapeutic targets, and especially if you want to regulate or modulate the immune response. And it's been discovered a long time ago, and many drugs has been targeting this. It also has multiple isoforms, so it's a quite interesting target. And there has been antibodies and small molecule drugs targeting this target. So bringing the nanobody for this target would be a very new, interesting venue. So how are we going to approach this? The overall workflow looks like this. So we will have our Strep-Tactin biosensor, Strep-Tactin XT biosensor, and the first step is actually quantitate. We wanted to demonstrate how we can quantitate these twin strep-tag nanobody in the solution, and we spike them into the E. coli lysate just to mimic how would you use it when you just express it and they're still in your crude lysate. And after loading these twin strep-tag nanobodies, and we were allowed them to interact with the CD45, which is our target in this case, and look at the target binding between the nanobody, and measured how tight the binding between the two is, which tell us how good your nanobody is. And so the main goal is to simply select the strongest binding nanobody against the antigen CD45. And in total, we received 32 recombinantly expressed nanobody purified in courtesy by IBA using their Strep-Tactin XT column. And all of them are purified from the E. coli lysate. Okay, so moving on to the first step, where we high throughput quantitate 32 nanobodies all in one run. So how do we achieve that? So experimentally, just a quick reminder, it's simply just take our sensor and a quick equilibration in the buffer and binding, and then dipped into your sample well. Now, because on top of 32 samples, we also need to have a standard curves. So in this case, instead of using 32 channels, I used the 3 columns of the channel, which also showcase that the biosensor, we don't really need to use full 32. You can use still 8, you can use 16, you can use any numbers of the sensor depending on your runs. So in this case, the first dip would be binding the standards, which is the 4 and the 5 column in the first plate. And we were able to fit 3 plates. So in this case, we only using 2 reagent plates. And of course, if you use 384 plates, you really can fit a lot of samples in one run. And after that, we will run the column 6, which is the first set of 8 unknown nanobodies, and then column 7, 8, 9 are the subsequent nanobodies. So from there, we will have 2 standards and then 4 columns of our unknowns. And you can see that overall, it's a very quick binding, right? Within 30 seconds, we can already see a very good signal response. And therefore, overall, you can see it's less than 5 minutes to just finish 32 samples, including the nanobodies and the standards and the unknowns. And also, just a quick reminder, these nanobodies are spiked into the 100x diluted E. coli lysate. So from there, we were able to get a very nice binding curve, and then we were able to fit them to figure out their concentration. That is because molecular binding is actually, the rate of its binding is proportional to its concentration. So the more concentrated you are, of course, the quicker you bind, just simply because more of them are present. And that is, we can use a kinetics model to fit this and to figure out the concentration. How do we do that? We have the standard curve. So you can see here, these two, and the rest of these are all standard curve. And all these samples are in between our standard curve. So as you can see here, we have the nanometer shift, which is just a unit to represent using the binding curve fitting to figure out what was the end of its signal. And from there, we can put them into plot them together in this linear correlated curve. And the blue dots represent the standards, and the orange part represents the unknowns. So from there, you can see here, we were able to obtain all the concentrations of these 32 expressed nanobodies. Moving on, from these quantitative nanobodies, we also need to figure out how good they bind, right? So that is the second part of the whole experiment setup, which is using the loaded nanobody on our probe, and we allow them to bind our analyte, and in this case is the CD45 antigen. How do we approach these typical type of K-off screening? Is first, we will have a baseline using our K-buffer. K stands for kinetics. It's optimized for kinetics analysis. And then we have our twin Strep-Tactin nanobody loading onto our tip. A quick baseline, and then followed by the association and dissociation of the antigen. In this case is the CD45. And you can see that this time I used 32 biosensors altogether, and that each sensor represents a nanobody. And of course, like Fabian mentioned earlier, these regenerations are totally possible with the sensor. Similar to their resin, our biosensor can also regenerate. So part of these sensors actually regenerated from the previous quantitation run. So all in all, within 30 minutes, we were able to extract 32 kinetics profile from these nanobodies binding to CD45, as you can see here. And you do get a range of different binding characteristics. For example, this nanobody, this one 1666 and then 1692 almost showed no binding, simply because of its the noise means overall the signal is very low. We really don't see much of a binding. But nanobodies like 1670 showed a quick association, a dissociation, showing it has probably not very strong binding pair. But some of the other nanobodies you can see here, the dissociation rate is very slow. So these could be the potential target. But of course, we want to know, we want to be able to actually extract the constant so we can compare them quantitatively. So first, we will fit each individual curve using the kinetics model. In this case, the 1-to-1 binding model. The residual plot shows how accurate the fitting is. And from there, we were able to figure out the K-off rate and the K-on rate and the overall KD constant from each expression. And from there, we ranked each individual nanobodies based on its K-off. So K-off rate, we want them to be as tight as possible, which means you want a small K-off number, and meaning that it takes really long time for them to dissociate, right? So in this case, I made a line of 10 to the -3 because normally molecules below that numbers are considered pretty tight. And in this case, we can see that we showed a few hits that passes our criteria. And these nanobodies are the one we're going to pick these nanobodies and to do a actual more accurate, detailed kinetics analysis. And how do we do this is actually utilize the steady state analysis, which is we will have a multiple titration concentrations of your antigen, and then we will measure the kinetic binding and dissociations of antigen at each concentrations. And in this case, similar assay procedure like earlier, we will have the loading of the nanobody, a quick baseline, and then the association and dissociation of the antigen. And antigen in this case, as you can see, we have 4 different concentrations, 0 being the background. So all in all, we have 32 sensors and we have 4 concentrations. So we can accommodate 8 nanobodies for a detailed kinetics analysis. So this is what the results looks like. We have 8 selected nanobodies, and then we were able to measure the each binding at different concentrations. And from there, we can then rank them again based on the detailed kinetics analysis. And then you can see here that we already have our 8 clones that are in the really good range. So I would say the top 3 would be, even top 4, they're below 5 nanomolars, would be a very possible lead candidate to move on to the second stage of your discovery research. And on top of that, you can see the steady state analysis, which of course you are welcome to add more concentration points to make the steady state analysis even more accurate. And on top of that, we have the iso affinity plot, which just shows the sort of the difference between the K on and the K off, because sometimes you might not just want the tight K off, you might want a quicker K off in this case. So here, depending on what you're looking for, you can also plot this K on and a K off to really narrowing down that few lead candidates that you're willing to proceed. So in total, we only use 32 biosensors, and we were able to do the quantitation, a quick screening, and to do a selected detailed kinetics analysis, all just within 1 hour. And that 1 hour is the runtime, right? So if you have the sample plates ready, you just need to put them into the Gator Pro system, and the machine will just run these analysis for you. So in conclusion, we showed that there are the few key technologies that are essential developed for the nanobody biotherapeutics discovery research. And the first part is this unique expression tag that are just suitable for nanobodies, and it's and great for kinetics analysis, and that is the twin strep tag and the Strep-Tactin XT. And because it's super strong binding between the two and reversible using its regeneration capability, it's able to achieve very cost-effective methods for high efficiency and capacity purifications for your purification needs. And of course, because the strong binding, we can use it for kinetics analysis on top of that. And it's sort of a one-tag solution for your research. And on top of that, using Gator Pro System and our designed Strep-Tactin XT biosensor, we are able to do the quantitation from the crew samples, analyze these nanobodies in a high throughput manner. And these, because of these proteins are regenerable, we were able to design the biosensors also using that regenerability, making the overall solutions very cost effective. And with that, I would like to thank everybody here today. And of course, Fabian from IBA for presenting a great purification technologies. And from our end, we showcased our Gator Pro system. And of course, feel free to reach out and we'll take questions from now. Thank you.
Thank you, Fabian and Johnny, for an excellent session. That's all the time we have today. If anyone submitted a question that wasn't addressed, keep in mind that the speakers will reach out to you directly. This session was recorded. You'll receive a notification in 24 hours when the on-demand session is available for viewing. And please take a moment to complete the feedback form so we can continue to improve your Digital Week experience. On behalf of Informa Connect Life Sciences, thank you and have a great day.